Okay, quick question. How long have you been alive? And no, I'm not asking you how old you are. I want you to really think about this for a second. This is a video about you at the smallest scales. My actual question here is, how long have the processes that make you, you, been working? When you come at this from the biochemical angle, the answer here can get a little hazy, and if you stick with me, we're going to land on a pretty wild answer. Because here it is, the real start of everything. This is your DNA being replicated. You have to hunt real hard to find a process more ancient and more fundamental to you and all other life on Earth. All your growth, all your motion, the entire business of you being alive is built on this foundation. We're looking at a complex of round about a dozen molecular processors working in perfect coordination just to make sure that one set of DNA in one of your trillions of cells can turn into two sets of DNA, and this is happening ahead of one small cell division happening all across your molecular landscape millions of times every second. This is your biochemical ground floor. Take the time to explore your DNA and pull apart the mechanics of DNA replication, and you'll come away with a pretty profound understanding of your place in this unbroken 3.8 billion year chain of life on Earth. It's time to explore the eukaryotic replisome, y'all. Let's get into it. And hey, welcome back to Clockwork. This is the first episode in a long series where we'll explore your DNA and how it powers everything you do. Subscribe so you don't miss a single episode. And I guess we should start with the, basics, right? So, what even is DNA? This is deoxyribonucleic acid, or, DNA. This helical polymer is how your body stores information. All the instructions necessary to build you live in your DNA. In order to carry out the business of keeping you alive, every single one of your cells needs to constantly synthesize proteins to do basically everything you need to do. The instructions to build those proteins lives in these DNA molecules. And we're not even going to cover how DNA does that work, right? We're not even leaving the ground floor here. Before your cells can carry out any of that work, your DNA needs to be copied first. Your cells are constantly growing and dividing to maintain, you. And so each and every time one of your cells divides, your cells have to completely copy all 3 billion base pairs of your genetic information. That helical structure helps DNA become extremely stable, but it hides the true purpose of this polymer. If we unwind the strand, we'll see DNA's real magic. DNA is a sequence of letters, like a sentence. These colorful bases encode information that your cells use to build, you. All the business of you being alive is hidden in these letters. What's really wild here is that you only get 4 letters to encode all that information, though. Your 4 DNA letters are adenine, guanine, thymine, and cytosine. If you look carefully, you'll see that adenine and guanine are kind of similarly shaped, with thymine and cytosine also looking kind of similar. These shapes make it so that these bases can bond with each other. Adenine can always stick to thymine, and it turns out that guanine can only stick to cytosine. It's always a short letter bonded to a long letter. And wow, there's some great symmetry here, actually. If you clean these shapes up a little bit, you might have yourself a pretty solid logo. Maybe something to stick on a hat or a shirt or something. Make a whole identity out of it. I don't know. Anyway, let's zero in on adenine to look at how this chain gets built. Adenine is held onto the DNA strand by this little ribose sugar and this phosphate group on the back end. Because of that shape, new letters can get stacked on top of DNA in a linear sequence. The real superpower here lies in how these letters always pair off. Since each letter only binds to one other letter, you can split these strands apart, add new nucleotides, and have 2 identical copies of your DNA with no errors. This is one of those little perfections in life. DNA is fine-tuned specifically to be copied. And that's where we are now, at the phase of cell division where it's time to copy your DNA and get ready for some good old-fashioned mitosis. So this is happening inside you at every level, effectively millions of times a day. Like I can practically pick a spot at random here and catch a cell in S phase. So let's meet our machines. How on earth can we copy the roughly 3 billion letters in each copy of your DNA at a time? It comes down to this incredible complex of molecular machines working together with incredible speed and accuracy. This is your Replisome, one of the most foundational and most powerful forces of nature. This thing looks like a beast, but we can assemble this thing one piece at a time and get a pretty solid understanding of how this all works. So let's just go ahead and jump in. At the moment your cell kicks off replication, it'll assemble the first piece of your Replisome right on this DNA strand. This is CMG Helicase, a brilliant little motor that unwinds your DNA. Up top here, we've got timeless tippin and claspin. which appear to help guide the parent DNA strand into the CMG helicase to help speed up the rate of replication. Once these are built, support structures like gins get attached to the CMG helicase. These stabilize the motor and actually kickstart the whole thing, letting it start pulling your DNA apart. If we go down to the business end of CMG, we can see kind of how one half of the parent DNA strand is getting blocked from entering helicase while the other one gets continuously pulled down. Over here is the lagging strand of your DNA. We'll get back to this complicated little goofus in a second. If we move down to the core of helicase, we can see how this little motor works its magic. Helicase is constantly using these little arms to walk this single strand of DNA down through the motor. And by the way, this strand also has a label. This is your leading strand of DNA. But if the DNA strands are complementary, why do we even bother with the distinction between the leading and lagging strands? You know what? That's called foreshadowing, folks. For now, we're going to follow the leading strand down through helicase to its next stop. In eukaryotes like us, the leading strand is pulled all the way down through helicase, and then, based on our current understanding of replisome structures, has a surprisingly long distance to travel before it hits the real business end of DNA replication. Down here is DNA polymerase, another one of the most powerful forces in all of nature. Specifically, though, this is DNA polymerase Epsilon. In school, you might have learned about DNA polymerase as a single enzyme. Well, that's the case for our prokaryotic cousins, who have replisomes that we have a way better understanding of. Over here in Eukaryote Town, we love making things just a little more complicated. We've got 3 DNA polymerase variants all working together to copy this genome, at least at the first stage. There's actually kind of a lot of DNA polymerases, and we're going to cover all of them across the course of this playlist. DNA polymerase Epsilon works exclusively on the leading strand and is where we're going to learn about the magic of DNA replication. As the leading strand is wound down here, it's going to come in contact with pole E. Right in front of pole E is our pCNA sliding clamp. Basically, when DNA replication gets kicked off, pCNA clamps onto the DNA strand right in front of pole E to help hold everything together. But let's move back to pole E's active site and check out how this DNA print actually happens. If I turn DNA polymerase on its side, you see it's got this U shape that perfectly fits DNA. But the main thing to keep in mind is that polymerase holds onto DNA in the exact right position to allow a complementary nucleotide to bind to the template strand. This little hole back here is a tunnel that only the next correct letter in a DNA sequence can fit through. And watch carefully, lone nucleotides are a little bit different before they get attached to DNA. Instead of just one phosphate on their backbone, these letters actually have 3 phosphates. As they come in contact with their complement letter on the other side of the chain, the other 2 phosphates will pop off, securing this letter onto the growing DNA strand. Breaking these 2 phosphate bonds releases a ton of energy, which helps push the whole polymerization process along. And you know, if that sounds familiar, well, it should. I mean, just check back with adenine over here. Adenine plus 2 extra phosphates is just adenosine triphosphate. You know, ATP, the energy currency that your body uses to power literally everything you do. Your body literally repurposes DNA letters to move energy around your cells. I just love that little bit of symmetry. Nothing goes to waste inside your cellular landscape. Heck, even guanine gets in on the action by being an important energy carrier for transmitting signals around your cells. But that's something we need to save for another video. And for the leading strand, that's pretty much it. That's all that really happens here. DNA printer go burr, no drama. Awesome. Technically though, we're in the middle of the replication process, and at the very start, this leading strand needed to have an RNA primer attached to it so that pole E had something to latch onto and extend. But we're about to see that play out like dozens of times with the lagging strands, so we don't need to talk about it right now. Instead, let's buckle up and talk about how this gets super complicated. The way this replication fork is oriented is perfectly tuned for the leading strand. Just feed more DNA in, and pole E can just churn out more nucleotides with blazing speed. And so, for the lagging strand, I mean, we're just adding more DNA letters onto it as well, right? Why have I been, foreshadowing how complicated this is? Let's go ahead and go back to our original models we used to show how new letters get added onto a DNA molecule. Like I said, it's super easy for DNA to polymerize. Just get the backbone sugars in contact and boom, they bind. Easy. But what if I try to add new nucleotides from the other direction? They're not going to attach. Remember, we need to break those 2 phosphate bonds to power this reaction, so in a way, the opposite end of this rung is the glue we need to polymerize DNA. These letters have glue only on one side. You can't just flip it around and attach from the other side, which means the DNA can only be built in one direction. That's why each end of the DNA molecule has a label. This side is the 5' and that's the 3' end. DNA can only be synthesized from the 5' to the 3' direction. Are you starting to see the problem? We can only build DNA in one direction, but these two DNA strands flow in opposite directions. So to bring this all together, Helicase is moving the Replisome along this DNA strand from the 3' to the 5' end. This is great for Poly, right? Because the DNA is flowing in the right direction for them. They're just going to keep churning out new DNA with no drama. However, up here, the lagging strand is basically moving backwards, entirely in the wrong direction. The Replisome is pretty clever here. We can get around this catastrophic misalignment. We're just going to build the lagging strand in fragments. So let's just get into it. As the lagging strand gets kicked out of Helicase, it's going to hit this beautiful subunit called the Primosome. This is technically two different molecular processors. We've got primase over here and DNA polymerase alpha right here. Like I said before, we can't synthesize new DNA without something for the polymerase to hook onto. That's what the Primosome builds. The Primosome only needs to do this one time on the leading strand, but it has to continuously do so on the lagging strand every 200 nucleotides, give or take. The process is pretty wild here, and I am shaving a bunch of details off to keep this video from going on way too long. But basically, as the lagging strand exits Helicase, it will run right into the business end of primase. Primase will glue a small 10-letter RNA primer onto the lagging strand and then disengage, allowing polymerase alpha to swoop in and extend this primer by about 20 more letters before it too pops off. So after the Primosome does its business, this little section is going to spool out about 200 nucleotides downstream from the Replisome before our final contributor shows up. This is polymerase delta, the lone wolf DNA replicator. Pol-D is loaded onto the lagging strand one primer at a time, synthesizing the lagging strand in the correct direction until it hits the next primer. And then it gets disengaged, allowing the cycle to repeat for each little fragment of the lagging strand. And if we take a deep breath and pull back, we can see this whole process in action. The leading strand is synthesized in one fluid reaction while we have all this machinery over here to put the lagging strand together 200 letters at a time. But what we're left with is a complete and accurate copy of your genome. And if this feels exhausting, no big deal. This only happens every time your cells divide, which means it's happened in your body about 20 billion times over the course of this video alone. You know, give or take a few billion given your age and whatever. And incredibly, this all happens with very few mistakes, all in perfect time with the rest of your cellular growth processes. It is so small, yet such a staggeringly large process. And that's what I want you to think about. This little loop has played out endlessly across all your cells every single microsecond that you've been alive. But the question is, when did it start? Or maybe a better way of thinking about it is, does it ever stop? And that's really the question I want you to ask now that we're thinking at the biochemical level. Because the argument I want to make here is a bit of a reach. How long have you been alive? When did your DNA start replicating? Well, I mean, replication can pause depending on what's going on in the cell cycle, but as long as you're alive, it never actually stops, right? This replication process is genuinely endless, and this very physical process happening in you right now has been going on for way longer than you've been alive. Everything else on this channel, every other part of biochemistry, is just a way to help keep this story, this DNA replication process going. And if you really think about the totality of it, let your mind reach out and try to connect to the actual scale of what the endlessness of DNA replication means, your DNA dividing right now is just the latest iteration of a story and a process that has gone on unbroken for a single instant for nearly 4 billion years. And sure, maybe you can push back and say that your initial set of DNA was created whole cloth from your parents, but in reality, you got 2 sets of chromosomes from your parents that were created through a process similar to DNA replication. We think about DNA in the abstract so much that we forget it's a physical object. The DNA that eventually became you was replicated and then stood waiting until the moment that it combined and, began the process that is now you. It is still a part of this nearly endless chain of DNA replication going back all the way to the dawn of time. DNA replication pauses, but it has never stopped. You are not separate from the whole history of life on Earth. 3.8 billion years ago, somehow this process appeared on Earth, and DNA started replicating itself, and it has never stopped. If you understand all the ins and outs of DNA replication, it gives you a little window into your billion-year history. You aren't an island. You're the latest link in an unbroken chain that reaches back across the whole history of life on Earth. There is a tangible line that connects you watching this video all the way back to the very first time the original cells started replicating their genes at the very dawn of life on Earth. You're standing on top of a foundation billions of years in the making, and all the while your replisomes are dutifully copying your DNA. That is a constant chemical little mantra, your cells copying your code over and over again, constantly whispering all over you, I am, I am, I am, I am, I am, I am. And so you are. There's so much more to explore here. We haven't even scratched the surface of how your DNA powers the work of keeping you alive, but this is a good foundation to stand on for now. I really appreciate your time. Thank you so much for watching. And again, thank you so much for watching. If you like this video, please consider subscribing to this channel. I'd also appreciate it if you checked us out over at watchclockwork.com or over at our Patreon as well. Find all those links in the bio below. Feel free to sign up for our newsletter, which is going to be a weekly exploration of all the biggest news in biochemistry. That honestly helps the channel quite a lot. Either way, we've got new episodes on the way monthly for now, but we're slowly ramping up with our new style. If you like the new style of Clockwork videos, if you like us going 3D, please leave a comment. I'd really appreciate any feedback you have. Otherwise, I really appreciate your time. And as always, we like to leave you with peace, love, and nucleoside triphosphates. Everyone be well. Thank you so much.